divide. In this last video for chapter 2, we're going to go over cell growth and division. We had briefly mentioned some of these things, talking about the cytoskeleton in the last video, but now we're going to get a little bit more detailed, and hopefully this, at least to some degree, is some review for you. So by the end of this, you should be able to talk about the stages of the cell cycle in their proper order, talk about how the cell cycle is regulated, what can happen if the cell cycle loses control, and then we're going to talk about mitosis and cytokinesis in their proper order. Now, before we get too far into it, we have to make some definitions for ourselves in what types of cells we find in the human body, because there's really two major types. Ignoring, things like muscle, heart, like blood, all that kind of stuff. But there's two major lines of cells: somatic cells and germ cells. Somatic cells are your body cells. These are cells that make up your skin. You find them in the liver, you find them in your muscle, right? They're just kind of like, if you're thinking of a cell, the somatic cell is probably what you're thinking of, because it's pretty much any cell you could find in your body. We have the 23 pairs of homologous chromosomes. Remember, homologous means they're similar but not exactly identical. So they have the same type of information but not necessarily identical information, because you got one set of these chromosomes from your mom and one from your dad. If they were identical, that meant your parents were identical, and now we have questions. So we have homologous pairs of chromosomes, 23 pairs in the adult human, with 46 total. This means we are diploid, so you'll see that term diploid, meaning you have two sets, right? Two sets of chromosomes. And so these are your somatic cells. So every single one of your somatic cells, every cell that makes up your skin, your liver, your muscle, right, are all diploid cells that have these 23 pairs, one set of pair, from the pair coming from your mom, one set from the pair coming from your dad when you were conceived. And then the germ cells. The germ cells sometimes are called sex cells, because these are the cells that are involved with sexual reproduction, eggs and sperm, sometimes also called gametes. So these germ cells, are not the same as the somatic cells, because instead of having the 46 total chromosomes with the pairs, the germ cells, the gametes that we form, like the eggs and sperm, have half of the number of chromosomes. So they only have 23, and there are no pairs. Reason being that if you are an egg, the hope is that a sperm will come and fertilize you, bringing along its set of 23 chromosomes. So if the egg has 23 chromosomes, the sperm has 23 chromosomes, when they come together in the form of fertilization, then we will be back up to 46 with pairs, right? If, let's say, an egg were to have its full 46, if it were to be fertilized, we would have literally 50% more of chromosomes than we would need, and it would not likely be compatible with life. So now let's get into the cell cycle. And again, we're going to focus on the somatic cells. We're not going to get too deep into meiosis. We're going to mainly focus on just the generic cell cycle, just everyday life for the cell, and then how the cells divide, if they were like your muscle or your skin or your liver cells. So the cell cycle is a very well regulated process your cells go through, where we have a life cycle. Basically, we go from doing our normal daily jobs and then dividing to create more cells. There's two main sections of the cell cycle: interphase and mitosis. Now, we can break interphase down into multiple parts, which we will, and same thing with mitosis. We can break mitosis down into multiple parts, which we will, but these are the two main divisions: interphase and mitosis. If you think of it, interphase, our cell is not dividing; mitosis, our cell is dividing. So let's talk about interphase first. Interphase, we are not dividing, and most of our time as a cell is spent here, about 90% on average. This can vary depending on the type of cell we are looking at. So don't look at the 90% and think that's a hard and fast rule. It can vary, but if you were to be asked a question, I would generally put 90%, right? And if you put that on an exam, that would be completely fine. So cells spend most of their time in interphase. And there's three parts to interphase, and each of these has an important um, job or role with interphase. We have the G1, the S, and the G2 phases. G1, also known as gap 1 phase, is the first one, and this is where we do some growing. So we're going to do some growing, we're going to be kind of doing some of our normal day-to-day activities, and then we get to S phase. S phase, or synthesis phase, this is where we are going to replicate all of the DNA. Make sure you remember this. I definitely asked this question in classes like Bio 5, and so I expect that you should be able to tell me this in anatomy as well. That S phase is where DNA gets replicated. We do not replicate DNA in mitosis. Trying to replicate DNA in mitosis would be like trying to write your essay as you were submitting it. Wouldn't work very well, right? So think of this S phase where we're replicating the DNA is like when you write your paper before the deadline, before you turn it in. So S phase, we replicate the DNA. G2, this is our second gap phase. This is where the cell's going to continue growing. It's going to make all the necessary preparations for mitosis. So G1, we're going to kind of do normal day-to-day stuff. We're going to double check and make sure there's no damage to the cell, make sure the DNA is intact. S phase, we replicate DNA. G2 phase, we're going to do some more growing, make sure we have all the organelles we need for dividing, make sure we have any other prep work done, double check that the DNA replicated properly before we go into mitosis. There also is a G0 phase, which is a resting phase, and usually you'll see this G0 phase branch off from the circle that forms the cell cycle. So here we go. So here is our G1 phase, right, in this first one here on the right, S phase, and then G2. And you notice the mitosis sliver, the M sliver here, is really small. And that just kind of is to demonstrate that we don't spend much time there. And then G0, we're not actively dividing, and so we're just kind of going off. We're just not focused on trying to divide. And you'll notice we can go into G0 for long periods of time and then go right back into G1 if necessary. So DNA is replicated during the S phase. So we go from having one copy, right, of each of those 46 chromosomes. Now every cell starts with 2 copies of the 23 chromosomes, so we have 46. Now we're going to make double the copies. So we have 46 duplicated chromosomes at this point. The copies are referred to as sister chromatids. Right, so sister chromatids are 100% identical to each other. And I do want to make a clarification here. I realize the slide has a little bit of confusion. So we started with 23 pairs. We have now duplicated all 23 pairs. So we had 46 chromosomes, individual chromosomes. Now those 46 are duplicated. Right? They're duplicated, meaning each chromosome is now composed of 2 sister chromatids that are held together at a pinch point called the centromere. Right, so here's our homologous pairs, right? So this would be one sister chromatid in red here on the left. This other one is the other sister chromatid on the right. These 2 red ones are 100% identical. The blue ones behind them are very similar because they're homologous to each other, but the blue is not identical to red. It's very similar, same basic information, but not identical. The 2 blue lines here are identical to each other. So we look at what's going on in terms of dividing. We've gone through interphase, now we can go into mitosis. Mitosis has a few basic phases. Now, depending on the resource you read, it can be 4 or 5, right, plus cytokinesis. Some textbooks will combine prophase and prometaphase together. For us, right, we see that division. And so just keep that in mind. Some resources have them split into pro-meta, meta, ana, and telophase. Some books will only say pro, meta, ana, and telophase. So just keep that in mind. It's not that a resource is wrong or outdated, it's just that some books prefer to go 4 phases, some prefer to go 5. For us, we're going to go 5 phases. So we have prophase. This is where we are going to see chromosomes condense down and become visible. The DNA inside the nucleus goes from being a jumbled mess to now distinct chromosomes that you're starting to be able to tell apart. The centrosomes, made of the centrioles, are starting to form spindle fibers. The nuclear envelope is breaking down, so we're going to remove the boundary from the inside of the nucleus to the outside. And the centrosomes are moving to opposite sides of the cell to form the poles, kind of like a north and a south pole. So that's what's happening in prophase. In prometaphase, we're continuing on, and so the chromosomes continue to condense down. Now they're very visible. That nuclear envelope is basically gone. And the spindle fibers that were being created by the centrosomes are now reaching out and grabbing onto the chromosomes. In metaphase, we have those chromosomes locked onto those spindle fibers, and we have basically played a game of tug-of-war between the two poles to line these chromosomes up along the middle. This middle line that we're lining up on, sometimes you'll hear it called the equator, sometimes you'll hear it called the metaphase plate or the metaphyseal plate. Generally speaking, I'll just use whatever is in our textbook, right? So metaphase plate is fine, but if you say equator, I'll know exactly what you're talking about. So each sister chromatid is attached to each side. So if we look at this chromosome here, right at the top, we have the 2 sister chromatids. The sister chromatid on the left is attached to the left pole. The sister chromatid on the right is attached to the right pole. And right now, since they're still locked together, it's like a game of tug-of-war that no one's winning, right? It's a game of tug-of-war that's currently tied. Then we go into anaphase, and now we have released the sister chromatids from each other, so we have severed that binding between them. And so our sister chromatids are going to fly towards the opposite poles. So this sister chromatid that was attached to this pole is going to get pulled over to the left. This sister chromatid attached to this pole is going to get pulled to the right. So they're separated from each other. And so this is how we end up with the same set of DNA on each side. Because remember, the sister chromatids are 100% identical to each other. So what we're doing here is we're creating a side of the cell that has a copy of the first, second, and third chromosomes, right? And this side over here also has a copy of the first, second, and third chromosomes. Perfect. That means that the cell that we started with is going to be identical to the cells we end up with. And then telophase, I like to think of telophase as like the opposite of prophase. A lot of the things that happen in prophase, we're just doing the opposite. So if you remember, prophase, we saw the nuclear envelope break down. And in telophase, the nuclear envelope builds back up. In prophase, we saw the chromosomes condense and become visible, and on telophase, we see that decondensing and that less visibility of individual chromosomes. The spindle fibers were building in prophase, and in telophase, the spindles are breaking down. And then we get to cytokinesis, and this is going to be where we actually split into 2. So it's important to note, prophase through telophase, we are just dividing up the DNA, right? We're just dividing up nuclear material, but we have not actually split. And it's important to note that while most of the time cytokinesis and telophase kind of happen together, it's not always required. This is one of the reasons why your muscle cells have so many nuclei. They go through prophase through telophase without the cytokinesis, right? So keep that in mind. Most of the time, telophase and cytokinesis do go together, but it's not a requirement. So that when we have a multinucleated cell, like a muscle fiber cell, we've actually gone through mitosis without the cytokinesis part. We've actually gone through mitosis without the cytokinesis part. So cytokinesis, this is where we're actually splitting the cell into 2. In plant cells, we would use something like a cell plate to create new cell walls. We don't have to worry about that. I'm not going to test you on that. The one thing you should know is how an animal cell does it, which is using actin filaments, which are part of the cytoskeleton, to form, kind of wrap around the middle there to tighten down. We call this the cleavage furrow because we're seeing this furrow, this like dividing wedge form, right? That is going to split the cell into 2. So basically we're winding these fibers around and around and around on the inside of the cell until we wrap them so tightly that we pinch into two. Now, the cell cycle does not mean we're just kind of going through it with no checks and balances. The cell cycle is very well controlled. There are checkpoints. And this is to make sure we don't have any errors and that it's the right time to move forward. Right? Because mistakes, if we pass those mistakes on, they can get stuck where we can't fix the mistake after we go past a certain point. We also want to make sure that our cell is actually receiving signals telling us to go forward, or if it's telling us to stop, that we've stopped. The main molecules that are going to help with this are called cyclins, and another molecule that works with these cyclins is called a cyclin-dependent kinase. And this is going to basically, these two are going to work in tandem. They're going to work together to allow us to go past certain checkpoints. If these come together at the right time and the conditions are correct, we can move through these checkpoints. If we lose control of the cell cycle, this generally results in cancer, right? It can be a reason why someone develops cancer, that the cell cycle has lost control. Think of it like your car, right? When you drive your car, you have a steering wheel that tells you where to go, and you have two pedals. You have a brake and you have a gas pedal. And the combination of you using brake versus gas keeps you moving in a safe manner, right? So that you can get to your destination safely and on time. If you lose control of your gas pedal or you lose control of your brake pedal, you're probably going to get in an accident, right? So you think of the cell cycle the same way, in that we are going through the cycle, and there's some places where we need to slow down or stop, and other places we can speed up. As long as we're following those rules, we can get to our destination safely. But if we lose control and we go too fast or we can't go forward, then we may end up with something like cancer. So here is control the cell cycle. Now for this class, I'm not going to make you memorize the combinations of cyclins and CDKs, but you should at least have an idea of what those combination names mean, right? And that these combinations are going to help us go past checkpoints as we come to them. But we're going to talk about three major checkpoints and the basics of what happens at each. So in the G1 checkpoint, right, this is where we are going to double check. Is the cell in good, healthy condition to potentially divide, right? That's kind of what we're doing there. We're double checking that the DNA is not damaged. We're double checking that the cell is in good enough condition to divide. Let's say there is some damage to the DNA, we might be able to fix it. And so the checkpoint gives us that pause to fix any errors that can be fixed. If it is too damaged, beyond recognition, beyond repair, then we can trigger apoptosis. Remember, we talked about that at the beginning. So checkpoint 1 is focused on making sure the cell is intact and without damage to go through the rest of the cell cycle. We get to G2 checkpoint right up there at the top. G2 checkpoint comes after DNA synthesis. So one of the major things we're doing at this checkpoint is we're going to double check, did the DNA replicate properly? Do we have 2 entire copies in their entirety that are undamaged? That's what we're looking for there, as well as any last little minute prep things before going into mitosis. And then the last checkpoint, we have the M checkpoint. This is going to happen during metaphase, like before, in metaphase before anaphase, because what we want to do is we want to double check the spindle fibers and make sure that the spindle fibers have attached to the sister chromatids correctly. That way we don't end up with sister chromatids going the same direction, because if that were to happen, one daughter cell would have 2 copies and the other daughter cell would have 0. So we're going to double check, did everything attach properly? Because once we split, right, once we cause that split to happen, it's too late. We can't go back and repair it after the fact. So that's why these checkpoints are so important, because once the cell divides, any mistakes that were not caught are stuck, right? We can't go back and fix them. Kind of like if you're writing a paper, you can go back and proofread it as many times as you want, but once you hit submit and it's submitted and the due date passes, you can't fix those mistakes that were left behind in your paper. They're there, they're locked in. Same kind of thing. We want to double check for all these mistakes that could potentially happen before we hit submit, right? Before we divide into 2 cells. And in this last section, we're going to get into differentiation, because so far we've kind of talked about a generic cell, but we haven't talked about, like, how do we get from a generic cell to these special cells? Because if you look at cells under a microscope, and which we will, you'll notice that our cells look very different from each other. You know, the cells we find in our skin don't look like the cells we find in our muscle. And that may lead you to ask the question, why? Or how? How do we get there? Cell differentiation is the answer to that. So we're going to talk about how we go from a general cell during development, like as an embryo, and stem cells that we find in our own bodies too. We'll talk about some stem cells as we go through the course. We're going to talk about how we go from general to specific. And then we're also going to talk about different categories of stem cells, because not all stem cells are created equal. Some are only found during certain parts of your lifetime, and others we find all the way through until the end. So let's talk about a stem cell first. Stem cells, right? And you may have heard stem cell before, and heard people maybe saying, like, this is going to be the thing that lets us move forward in treating lots of different diseases and conditions. You might also hear people talk about them as, like, that's horrible and not moral for us to use. You may have heard the arguments, but not fully understood what a stem cell is. So we're going to talk about that first, because stem cell is a generic term for an unspecialized cell that can divide without limit and can become specialized, right? So that's kind of the generic definition of a stem cell. It's a general cell, no special functions or anything just yet. It can divide a lot, right, without really any real limit, so it can just keep dividing forever. And if it's given the right conditions, it can become a specialized cell. Not all stem cells are created equal, though. There are different categories and different types of stem cells. We have totipotent, pluripotent, multipotent, and oligopotent. Those are our 4 categories of stem cells. So the different types of stem cells we have: totipotent, pluripotent, multipotent, oligopotent. Totipotent means it is a stem cell that can become any type of cell. Any type of cell. Not just in the body, but also to form parts of the placenta. So totipotent, this is the cells we would think of when we think of in the very, very beginning. I mean, we're talking fertilization and implantation has just happened. These cells can become the embryo, or they can become part of the tissues that support the embryo. Right? So they can become any cell, including cells that don't end up being part of the actual person. Pluripotent cells are usually the ones we're thinking of when we think embryonic stem cells, because these ones, the pluripotent cells, the stem cells, can become any cell within the embryo. So they can become skin, they can become muscle, they can become heart, they can become blood, right? They can become any of those, but they cannot become part of the placenta. Multipotent stem cells are a bit more limited. They can become a variety of different cells, but not all, right? There's a category of different cells that they can become. And then oligopotent, that's getting a little bit more specific. They can become a variety of different cells, or at least 2 or more different cells within the same type of tissue. We also have another one listed here called unipotent. Unipotent can just become 1, right? So it's just like an immature cell. So these are all different types of stem cells. So you'll notice the definitions of them, they can really vary depending which category we're looking at. The general process is the same, that we start with an immature, unspecialized cell and then differentiate and become specialized, right? That process of becoming specialized is called differentiation. And that's going to come down to, we're not going to get into the nitty-gritty, but that comes down to some specific genetic expressions that are happening within that cell. So certain things are happening on a genetic level and in the expression of certain genes at certain times that's going to lead to different cell types, right? If you're really interested in that, you can ask me. I'm not going to get into details because I don't want to go further than we need to. So let's take a look at one of these multipotent stem cells. In this case, we're looking at hematopoiesis, or hematopoiesis. And here we're looking at a multipotential hematopoietic stem cell. So we're looking at a stem cell that has the potential to become any sort of blood cell. This multipotent stem cell can then specialize a little bit to either a common myeloid or a common lymphoid stem cell. We then become a megakaryocyte, an erythrocyte, a mast cell, or a myeloblast if we're on the myeloid progenitor side. The megakaryocyte is what leads to thrombocytes, which would be your platelets. The erythrocyte, your red blood cell. The mast cell, which is already done, and the myeloblast can become any of these white blood cells you see down here, which would be basophils, neutrophils, eosinophils, the granular white blood cells, or a monocyte, the monocyte which can then further differentiate into macrophage or dendritic cell. On the common lymphoid progenitor, we can become a natural killer cell or a lymphocyte that can become either a T or a B lymphocyte. The B lymphocytes can then further differentiate to become plasma cells or memory B cells, which then hold on to the memory of how to make antibodies. We're not going to get into all these different cells and what they mean yet because we're not there yet. We have a whole chapter on blood, so we'll get there when we get there. But you see this table, just looking at the table, you can see how you have your multipotent cell and how many different cells this single cell has the potential to become. If we count it up, right, either cells or cellular components, we have 1, 2, 3, 4, 5, 6, 7, 8, 9, 10 different possible cells we can become. So we think stem cell, and usually people are thinking of like an embryonic stem cell, which people may be opposed to using, but this cell in this case, this is a stem cell we actually find in adults, right? This is something that you use in your body on a regular basis to make more blood cells, to make more, to replace the ones that are damaged or lost. I mentioned earlier in this chapter that erythrocytes or red blood cells have a lifespan of about 4 months. That means you need these stem cells to make more and more and more of them because you're having to go through and make them, 3-4 times a year. And so then if we're looking at, the process, and again, this is getting into the definitions of the ones we talked about earlier, the totipotent embryonic stem cell can become anything, including parts that are not part of the embryo, that are not part of the embryonic disc. The pluripotent embryonic stem cells is usually what people are referring to when they're talking about stem cells, right? We're talking about the cells that make up an embryo in the early stages of development. Pluripotent embryonic stem cells have the potential to become any type of tissue in the human body. The multipotent stem cells that come from these pluripotent embryonic ones then are getting a little bit more narrow, right? They can become, in this case we see our endoderm line ones can become anything made from the endoderm, so like glands and inner organs like the lung and the pancreas. The mesoderm line become things like blood and the heart muscles, right? Whereas the ectoderm line, that's going to be the outer linings like your skin as well as your nervous system, so neurons. And so when we're looking at adults, stem cells, we're looking here at the multipotent stem cell lines. But again, you have them. You have these to regenerate cells that need it throughout your lifetime.